All right. So first thing I need to do is to get this thing to work. All right. Is to get my conflict of interest out of the way. So some of the algorithms that we developed have been licensed to a company called Darwin Health. So you should take that into account. And Columbia also has an interest in it. So since the theme of the workshop is on mathematical analysis, I thought I'd start with a philosophical analysis instead. And this is based on the fact that we think of cancer as a highly dysregulated state of the cell. And in fact, I've asked a lot of people, if you do a principal component analysis of 100 cancer-related tissues from the same subtype of cancer versus 100 normal tissues from 100 different individuals, what would they look the same or different? And most of them say that the cancer should actually look much more spread out. And that's actually not true. Cancer is as tightly regulated as normal tissue. And in fact, when we look at these plots, we look at things like the sort of principal component analysis for different tumor types. In this case, for instance, this is cancer and this is normal tissue, cancer, normal tissue, etc. We have the impression that the cancer cells are occupying a substantial area of that plot. Okay, but you have to think that this is a two-dimensional manifold in a 24,000 dimensional space. Okay? And so actually, the amount of that space that the cells are occupying in terms of what state they can achieve is minuscule. Just to give you a sense, out of the 2 to the 20,000 possible states a cancer cell could be in, typically 2 to the 20,000, just to give you a sense, there's 2 to the 400 atoms in the universe. So 2 to the 20,000 is a pretty large number. Out of those 2 to the 20,000 states that a cell could be in, if you assume that genes are simply on or off, which is obviously an ultra simplification, the cell is probably in a few thousand different cell states. Okay? So what I'm going to argue is that in order to do that, a lot of the cellular machinery must be actually dedicated to maintain and preserve that tumor state. Practically, in normal cell physiology, what this is doing is preventing one of your liver cells, one of your hepatocytes, to turn into a B cell, or one of your neuron from turning into something else. But in cancer, you have a very similar type of logic, and even though there is additional plasticity, as Peter has shown very beautifully, and I'll show some other examples, it's still tremendously constrained compared to the number of potential states it could be. And this is actually very visible because if you see what happens here in TCGA, if you cluster TCGA by gene expression profiling, you'll see immediately that there's quite a number of very large bucket of profiles that look very, very similar. So for instance, these are triple negative basal-like breast cancer. These are basal-like bladder cancer is actually quite close, luminal breast cancer, etc. And as I said, I'll show here, these profiles are essentially clustered as much as a normal tissue would be from the same tissue in different individuals. So if you actually then look at the genetics of these tumors, well, boy, that's all over the place. So if you look actually at what's happening for triple negative, the genetics of triple negatives, in fact, they're called triple negative because they have none of the major genetic alteration that we tend to see in other type of tumors, type of breast cancer. Like, and so what's happening here is that virtually no two patients have exactly the same set of driver mutations. And in fact, for many of the patients in that cohort, actually driver mutations are not even well defined. We don't even know what is the recurrent mutation that actually drives those tumors. So I think there's a fundamental paradox that we need to sort of account for. That's where the philosophy comes in. And that paradox says that how is it possible that something so extraordinarily heterogeneous as the genetics of a particular tumor in some way coalesces on a state that is as tightly regulated and controlled as that of a normal cell? Okay. I don't know if you have ever thought about this, but it's a pretty interesting problem to think about, in my opinion. So the solution that we came up with, and it's been for a while a hypothesis more than a solution, but now I think we've done enough publications in this area demonstrating that this is actually true for many, many different tumor types, is that, in fact, one possible way in which this could be accomplished is if we had the mutational landscape of the cell essentially being upstream of a relatively small number of proteins that are responsible for integrating the aberrant signals coming from the mutations, and then downstream of those implement the transcriptional state of the cell through a bunch of other regulatory pathways. We have dubbed these proteins master regulators, because, and actually they don't work individually, they work as a module. In fact, one paper that just is just in press, was accepted yesterday at Cancer Discovery. We actually demonstrated the entire logic of the entire module for neuroblastoma, about 10 different proteins working together. And actually what happens is that these proteins, because they don't work together and because they have to maintain stability, are very tightly regulating each other. So they're literally a not independent set of proteins. They create a state of the cell and they maintain the state of the cell. So why is this happening? What's the actually the rationale by which we had to go through this kind of architecture in order to implement the phenotype? Because the same way that if you have an image and you want to preserve it, okay, the best way to preserve an image is actually to encode it into a hologram. Because I can shoot an image or I can damage an image and I will have lost that set of pixels forever. But if I actually encode it in a hologram and I shoot the hologram, that simply causes a little bit of a fuzzier image. There's a little bit of loss of fidelity, but the image is pretty much the same. Okay? So what our interpretation of this is that the genomic of the cell, the genetics of the cell, is actually to some extent living in a space that is the hologram of what the actual transcriptional state of the cell is. And why is that the hologram? Because you have to implement a system that is incredibly fault tolerant, where no mutations in any individual genes can actually cause the organism or the cell to die. And so hundreds of millions of years of evolution have created a system that is extremely tolerant, where in fact to increase the height of an individual, you need a combination of many genes out of maybe 400 genes. And where to induce cancer, you may use combination of many, many genes out of maybe 800 or more than 1,000 potential cancer genes. And so I think this is very similar, so we've been saying this for quite a long time, but very similar to what Jonathan Pritchard has recently come up with in terms of the what I call the omnigene hypothesis. Everything regulates everything, right? So more or less, but there's still some influence. The interesting thing is that if you actually formulate things in this way, now you have a platform, a framework in which you can actually go and make sense out of this. So by the way, I wanted to cite just very briefly how we came up with this idea. The first hint that this was actually the case was from a study with Riccardo Della Favara on diffuse large B-cell lymphoma, where there were two subtypes of this disease, one very indolent called the GCB subtype, germ cell divide subtype, and the other one, a very aggressive one, which is called the ABC, activated B-cell subtype. And the only thing that was known at the time, very serendipitously, in fact, from Lou Stout's lab and Ricardo's work, was that NF-kappa B was, in fact, a dependency of the ABC subtype. Right? So with Ricardo, we thought, well, why don't we go and sequence NF-kappa B? Maybe there are mutations in some of the patient. Absolutely no mutations. So where could the mutations be? So I said, well, maybe they're upstream. And we were lucky enough that actually immunologists charted the pathways upstream of NF-kappa B for many years. And they were, and actually specifically in immunologically related cells, and B cells in this case. And so we actually looked, and lo and behold, there were about 15 different recurrent mutations that we could find by just sequencing about 100 genes. And those mutations invariably corresponded to a change in the activity level and the dependency on NF-kappa B. Okay? So that was kind of a aha moment for me, because basically what that said, that there was this convergence of all this entire mutational space. And in that case, we only look for single mutation. But now we know that, in fact, this integration occurs across many different things that happen simultaneously within these individuals, probably with an additive effect or an epistatic effect. And so the second piece of information that we got, and this is work with Antonio Iavarone and Anna Sorella, was that, in fact, we could identify master regulator proteins that were the proteins that, in fact, were not the one that were differentially expressed, but were the one that regulated the differentially expressed genes. And so we basically use regulatory networks to traverse the regulation of the cell and going from the gene expression layer all the way to the potential proteins. And in that case, we found just two proteins, CB beta, CB delta, actually three, CB beta, CB delta, and STAT3, that were all synergistic in regulating the mesenchymal subtype of glioma. And so this has become sort of a paradigm that has essentially guided almost everything else that we have done. And I just wanted to introduce you because I'm going to bring it then down to the single cell studies, but I wanted to give you a bit of a framework to interpret what you're going to see. The exciting thing about this is that this has actually now resulted in quite a significant amount of work, translational, because what we have actually proposed is that instead of actually drugging the single mutations, that is just one way to implement the dysregulation of this checkpoint, maybe what we should do is to actually drug the checkpoint itself, the entire set of master regulators that work together to prevent that state. Why? Because if you drug, for instance, EGFR in a tumor that is EGFR dependent, you will kill all the clones that are in fact dependent on EGFR, but there will be some subclones that have either bypass or alternative mutations, and those will feed into the exact same checkpoints and will completely bypass the drug, and therefore will actually lead to very rapid relapse. And this is what we see almost inevitably for every tumor that is actually not driven by a fusion event. Fusion events have actually shown to be particularly resilient and being really extremely interesting as potential entry point for therapy. But the others, almost all the time, you get, almost invariably, you get relapse. So if you actually drug the cell here, what happens is that there really is no way around it. You can't go around the checkpoint. The only way to do it is to actually reprogram the cell into a different state. For instance, going from an adenocarcinoma of the prostate to a neuroendocrine tumor of the prostate. And that happens, but the nice thing about it, and I'll show some data later, is that you're actually really constrained because the number of states you can actually go into is pretty limited. Okay? And so this has actually led to quite a large number of clinical studies and clinical trials where we have done everything from generating biomarkers to having studies that now have progressed to phase 2, combination therapy in breast cancer. This just opened up. We just got funded by both Lustgarten Foundation and the Price Family Foundation for N-of-1 clinical trials in pancreatic cancer and gastric esophageal cancer. We have been funded by the CureServe Foundation with Andrew Kang for pediatric sarcoma, and now we're working with several of the people here to implement the same paradigm in pediatric tumors. So this is pretty exciting because it is moving to the clinic, and some of the results, as I'll show you, are quite exciting. But the question is, this is philosophy. This sounds great, but how the heck do you find these master regulators? I mean, what's the actual mechanics by which you can find them? And so this is really relied on an idea that was, I think, originally developed in the lab, but very much in collaboration with Antonio, actually, initially, because the idea was that one thing that we have been able to do extremely well is to build a regulatory network that was inferred computationally using information theory from a gene expression data, a large subset of gene expression data. And then what we thought is that maybe we could use the actual regulatory targets of a particular regulatory proteins, both the ones that are repressed and activated, as a multiplex gene reporter assay that would tell us essentially whether this protein is activated or not, either enzymatically or from a regulatory perspective, without actually having to measure its activity. And the nice thing about Arachne is that it gives you networks, it gives you regulatory networks that are completely context dependent. So you would generate it for a B cell, you generate a different one for a glioma, generate a different one for prostate cancer. And so we've generated about 40 different models, and I'll show you how now we don't even need to create more models. Just create an algorithm that can integrate across all the ones that we already created. But if you believe that, then what happens is the following. What happens is that the activity of a protein becomes actually just implemented through the expression of its target. So all we need to do is measure the expression as regulated by those promoters. And the idea is that if you have cells that are in two different states, for instance, you could have a primary versus a metastatic disease, you could have a drug resistance versus drug sensitivity, any kind of differential phenotype you may want to consider. Now, if you observe something where in one of the phenotypes all these targets are up and all these targets are down, and in the other phenotype all these ones are up and these ones are down, now you know that this protein is highly activated in this phenotype. Okay? Not only it's activated because we have more of it, but it's actually activated because it implements the signature that you're observing in those tumors, which is very important. Essentially, it's a mechanistic implementation of that tumor signature. And so, as I said, we've done this for about 6,000 proteins, including one big surprise was this works really well for signal transduction protein as well, as I'll show you, even for surface markers. But basically 6,000 proteins in about more than 40 different tissue contexts, including both normal and tumor-related. So these are the two algorithms, essentially, are the staple of what we do. There are many others I'm not going to talk about much about today, but basically Arachne is the one that generates the regulatory network from a large collection of gene expression profile, and Viper, which was published about a year and a half ago in Nature Genetics, is the algorithm that can go from the gene expression to the activity of the proteins that regulate the gene expression using the regulatory network built by Arachne. All right, so let's see an example of how this works. So if I take all the breast cancer sample in TCGA, this is what I get. I will get essentially that if I try to do unsupervised clustering with it, without actually selecting genes, just with every single gene that I have. And by the way, this is the same picture you get if you actually select only transcription factor, so it's not specific to the gene that you select. But if you, in an unbiased way, select a bunch of genes and you do unsupervised clustering using those genes, what you get is a very fuzzy picture where, in fact, in that case, you can recover the basal subtype, but the luminal and the HER2 are the yellow ones, are all mixed in, the luminal A and luminal B are all mixed in, and there are some subtypes that are not quite clear what they are. And also, if you actually look at the genes that are the ones that are the best at distinguishing these different subtypes, they're not very important genes. You know, the estrogen receptor is there, but you see tremendous variability from patient to patient, the progesterone receptor there as well. But you see everything from white to red to blue, et cetera, et cetera. Very wishy-washy picture. This is one of the reasons why you can't really use RNA unless you use it as a large panel of genes to make specific predictions. But this is what happens when we transform that picture using the Viper algorithm. So now, not only you completely recapitulate the biology that is actually identified by Peru in a completely unsupervised way without having to do anything else. So you get your luminal A, your HER2 ER positive, HER2 ER negative, you get a gradient from luminal A to luminal B. But now the genes that you recover, for instance, for the luminal subtype, are all the ones that we already know. FOXA1, GATA3, XPP1, the estrogen receptor, and they are rock solid red across a large number of patients. Okay? Same thing for triple negative. Now, I guarantee you probably have never heard of these genes in the context of triple negative breast cancer. So we'll get to that in a second. But one thing I want to show you is this. If I take one triple negative and I compare it to the average of all the luminal, and now I take the top 10 most differentially expressed gene, and now I ask, where are those 10 genes when I do the same thing across the other 190 profiles that I have in TCGA? Pretty much all over the place. So something that was in the top 10 most differentially expressed gene could now be the 5,000th most differentially expressed gene in another subtype, right? If I do protein arrays, so these are PPAs, reverse phase protein arrays, it's actually even worse. The distribution is even wider. If I do Viper analysis and look at activity, that looks like a printing error there. That's actually a distribution. It's so tight, meaning that if you are in the top 30 most differentially active protein in one patient, you may be in the top 100 in another patient, but you're not even going to be in the top 500. Okay? And that's very important because it gives you a recipe that says, we may be completely wrong, and this protein may be completely incorrect. But if we are right, these proteins are actually conserved across a wide swath of the patients that have that subtype of the disease. And therefore, if you come up with an approach to treat the disease using those proteins, then it may be more broadly applicable than what you can do with mutations. All right, so let's see, first of all, whether this works. So we took the 40 master regulators for both, 16, I think, for 16 for luminal and 24 for basal. We silenced them using multiple hairpins in 3 different luminal, basal cell lines and 2 different luminal cell lines. And what you're seeing is that about 70% of them induce a greater than 2-fold change in viability, either in favor of the basal, so more reduction for the basal, or greater reduction for the luminal. And by the way, if you look at the luminal, GATA3, ESR1, FOXA1, you get all the things that you would expect. Okay? Now, the things that you get here, probably most of them, probably maybe with the exception of FOXM1, there's a little bit of literature on FOXM1 triple negative, but it's actually not very clear. But most of these genes you would actually probably never heard in the context of breast cancer. And I wanted to point out in particular 2 genes, BCL11A and FOXC1, that had absolutely no effect. Why am I pointing them out? Because similar to the work that we did with Antonio, this is the single most, and actually with Corinne in prostate cancer, this is the single most statistically significant synergistic pair predicted by the algorithm, right? And so, remarkable they don't do individually. Let's see if they do something together. So what you can see here is that if you actually co-silence FOXC1 and BCL11A, there's not a single living colony left in long-term colony formation. Absolutely no effect on the luminal cells, right? This is the second best pair. It's FOXM1 and PTG1. Very dramatic reduction in colony formation in the triple negative, actually increases proliferation in the luminal. So these are exquisitely specific. You may think, oh, FOXM1, that's just a G2 gene. You know, it's going to have the same effect everywhere. Absolutely not. When you combine it with PTG1, you get an effect that is incredibly specific just to triple negative. It does absolutely nothing to the luminal. All right. So we've done these analyses for every single patient in TCGA. One of the advantages of Viper is that you can perform these analyses on a sample-by-sample basis. In fact, I will show you in a few seconds on a cell-by-cell basis. And what we've discovered is that if you do this comprehensively, you find about 96 different subtypes, including very surprising subtypes. For instance, prostate cancer has been very challenging to identify in terms of subtypes using gene expression profile. As far as I know, the only subtype that's been identified has been for the neuroendocrine subtypes, which is a completely different type of tumor. There's no, there's no subtyping prostate cancer at diagnosis in terms of, for instance, in a way that correlates with Gleason score. Well, as you'll see, we'll find 7 subtypes of prostate cancer that completely co-segregate with Gleason score. But the more surprising thing was that we found, this actually, it's now updated, this is work done by Evan Paul in the lab that's actually really talented and now the chief bioinformatician in the lab. Basically, there are only 24 tumor checkpoints, meaning there are only 24 modules of master regulators that essentially explain every tumor that we've been able to look at in TCGA. And many of them, if you look at the one that I highlighted, are essentially things that we've already published in, for instance, neural glioma versus mesenchymal, in triple negative versus luminal, etc. Okay. So one thing that I promised you in the beginning was that this logic was responsible for integrating the actual genetics of the cell and may actually help you understand how this incredibly complicated genomic layer ends up inducing aberrant activity of these master regulators. So what we can do now, we use an algorithm called Bindi and now a newer version called Cindy, which essentially can identify all the proteins that are upstream of the master regulator and who, by just from a regulatory perspective, can affect the activity of the master regulator. So basically, we're dissecting all the signal transduction pathway upstream of the master regulators, and we only look for mutations in those genes. Exactly like we did with Ricardo for the pathways upstream of NF-kappa B, except that now it's done in a completely automated way, instead of manually. So this is what happens. We can do this on a patient-by-patient basis. So we don't need to look for mutations that are recurrent in a cohort. We can actually look at just one single patient. So this is what happens, is that you can see for every single tumor where we did it, if you look at how much of the genetics of those patients you explain, using the first and second, up to the first 100 master regulators, you saturate extremely quickly. And on average, between 15 and 25 master regulator explain virtually completely saturate the genetics of these individual patients. Right? And I'll show you a little bit more detail there. If you look at these gray bar, in some cases you don't see them because they're so flat you can't see them, but the gray bar are the null hypothesis. If you just choose master regulator at random from transcription factors. Okay? So you don't saturate at all. So let me show you an example for prostate cancer. In prostate cancer, we found 7 different subtypes. And you can see, for instance, the subtype 4, subtype 4 and 6 are striking in that they almost contain only Gleason 9 and 10 and 8, and they are really the most aggressive one. Well, the subtype number 1 is mostly made of Gleason 6 and 7s. So first of all, you can see that each one of the subtypes actually saturates, but with different mutations. So the mutations in each one of the subtypes, because they regulate different checkpoints, are different. And now this gives you the ability, for instance, of taking a particular checkpoint of interest. This is the FOXM1 CMPF checkpoint that we've actually published in Cancer Cell with Corey Bada-Shen and Michael Shen. And now you can look at all the upstream mutations that co-segregate with the activity. First of all, you see that the patients, all the patients in subtype number 4 are all here. All the ones in subtype number 6 are all here, subtype number 1 are all here. So there's a dramatic effect where that checkpoint is really not activated in the patients that have the better Gleason score. It's dramatically activated the one that have the worst Gleason score. Okay? But also, you can actually find individual proteins that have mutation that co-segregate with the activity of the checkpoint. And you can do this on an individual by individual basis. In fact, we've actually already experimentally validated with Corey and Albert Ides, who was the first author on the Cancer Cell paper, this 5-1, and we've shown that each one of them, when silenced, induces aberrant activation of the FOXM1 CMPF checkpoint, both in vitro and actually one of them also very significantly in vivo. If you look at a different checkpoint, you'll find different mutations. So for instance, 10%, absolutely no co-segregation of FOXM1 CMPF. Spectacular co-segregation with this particular checkpoint is the one that actually is the best for subtype number 1. Okay? So the mutational state of the cell induces its state through activation of a small number of proteins, and you can't mutate those proteins because if you do, the cell dies. These are so fundamental proteins that don't tolerate being mutated. For instance, NF-kappa B, why Why is an F kappa B mutated in the LBCL? Because if you mutate it, then the cell dies. All right. So this pointed out to actually a very simple approach we can use to design therapies for treating patients based on checkpoint activity, which we've now implemented in several studies, including a very large study here at Columbia called the N of 1, where we've already enrolled 120 patients. And the idea is simply you take every patient, you get a gene expression profile, you run the Viper algorithm, and now you get the top most aberrantly activated proteins and the bottom aberrantly inactivated ones. How many of them? We choose 25 because we know from the genetics that with 25, you pretty much saturate the genetics of the individual. So we know that essentially you need about 25 of them to explain why the genetics of that individual induced that particular phenotype. And now what we do is we simply collaborated with Peter Sims on a different technology than what he talked about today called Plate-seq, that was published in Nature Communications a few months ago, that allows us to essentially do gene expression profiles for about $20, where now we can it allows us to create gene expression profiles obtained from perturbation of cells that are specifically selected to recapitulate the master regulator of the patients. We've identified about 20 cell lines that recapitulate about 90% of all tumors in TCGA at better than p-value of 10 to the minus 5 in terms of master regulator overlap. And so now you can actually perturb these cell lines with any compound. So typically we do all FDA approved and all compounds in phase 2 essentially anything that is safe to give to a human being, plus a bunch of other things. And now you're looking for a compound that essentially completely reverses the activity of that protein signature. So all the bad proteins that were up are now shut down. All the good proteins that were down are now activated. And the reason this is possible is because, as I told you, those proteins don't work independently. They literally work as a module. So they're either switched on or switched off. And when you find the compound that hits the right entry point, that entire module will switch off. Okay? So we did this for now 120 patients. We generated about 45. We transplanted about 45 of them in PDX models, and about 25 of them are growing, and 7 of them we have already completed multi-arm studies using drugs. So we actually tested 34 drugs that were predicted by these methodology, OncoTreat that I just showed you, or another one called OncoTarget that just looks at druggable protein that are aberrantly activated. And both of them actually have just received New York State CLIA compliance certification, so they can be used clinically in a number of clinical trials. And what you're seeing is that out of 34 drugs that were hypothesized purely computationally using the signature of the patient, 20 of them induced objective response in these mice, either stable disease or significant tumor regression. And 4 patients actually were treated. I called them 5 because one patient had 2 tumors that had prediction that were completely orthogonal in terms of the drugs. And so we actually treated each one of those masses with the specific drug that was predicted for it, not together, but in rapid succession. And essentially 4 of the 5 tumors had quite striking clinical response. We had a meningioma that had essentially disease progression-free survival of more than a year after having progressed very rapidly on anything, any type of approach that they tried. We had a triple-negative breast cancer, which you're seeing here with significant metastases in the brain, and you can see the reduction in the metastases after about 5 months. And this patient had about 7 months of progression-free disease. And then we had a GIST, and this patient responded in both of the metastatic sites. One disappeared, one went necrotic pretty much overnight. And unfortunately, the patient had another abdominal mass that simply could not be biopsied. And it was also, unfortunately, in very poor condition at the time. And so that did not respond to the drugs, but the other 2 that we could biopsy and for which the drugs were predicted actually responded very well. And more interestingly, what we've seen is that when you actually take biopsy from the mice, actually these are true explants from the mice, at 3 hours after the 3rd dose, so way before the tumors actually has had a chance to be killed, we wanted to see whether the drug were actually killing by truly reversing the master regulator signature, or were actually killing by some other bizarre mechanism. And so this is the GIST sarcoma, the KRAS mutant GIST sarcoma. And what you're seeing is that 5 of the 6 drugs, in fact, all the 5 that worked, if you go back to here, you can see these are the 5 drugs that had a striking effect, starting with fludarabine, that had the strongest effect, had a very significant reversal because the red bars are the positive master regulator that now completely shut down in activity. This is an activity space. The blue bars are the negative master regulator that all been activated by the drug. And the one drug that didn't work was the chlorpromazine, which is a a neurotropic drug, and actually this one put the mice to sleep. They couldn't even be fed the drug. All right. So now I'm going to switch and tell you, so this is the framework. Now I'll tell you how we can use this in single cell, because I think this is a really exciting area. And also, I think a fundamental fruit of the work that Peter Sim has done here at Columbia by bringing some of this technology to bear, which has been transformational for us. So you've probably seen a lot of these papers where people show that they can actually take single cell, profile them, and then they can do clustering, and they say, oh, look, I'm seeing B cells and macrophages and neutrophils, et cetera, et cetera. The problem with that is that, in fact, it then becomes very difficult to delve into the actual biology of the single cell. Why? Because of a very fundamental problem that most people shrug under the rug, which is the fact that the vast majority of genes are actually dropouts. You don't see a single read for that particular gene in that particular cell. Okay? And so, in particular, the transcription factors that regulate lineages, these are the ones where you only have a few copies of a few molecules per cell, and so those are the first things to disappear, right? So why is Viper useful? Because in Viper, we're using a large number of targets of a protein to estimate its activity. So we don't use the expression of that gene or the corresponding gene at all. We just use the expression of the targets of that protein. And we have so many of them that when you actually reduce the depth of sequencing from 30 million reads to 10, 5, 1, 0.5, et cetera, while the gene expression correlation drops dramatically, so we are correlating the gene expression of the 30 million profile with the gene expression of, say, 100,000 read profiles, it drops like a rock. On the other hand, the activity profile that you get through Viper is essentially almost unaffected until you get to about 0.5 million reads and degrades very gracefully. And so when you get to the range of single cell biology, by the way, this is the range of plate-seq biology, where we can now profile at about 2 million to 1 million reads with virtually no change in activity profile correlation with respect to 30 million. But if you go to a single cell level, which is around maybe 50,000 to 20,000 read, you still have a correlation of about 0.8, okay, as opposed to a correlation of maybe 0.3 that you would get from gene expression. So this is great, but there's a bit of a problem. Single cells are like a box of chocolate. You never go what you're going to get. And so when you actually open the box, the Viper algorithm is dramatically sensitive to using the right interactome. If you don't use the right interactome for that corresponding tissue, you get completely random results. So how are you going to know which interactome to use for which cells? And so what we do now is essentially we first do the clustering, which works well even for everything else, and we then generate a network using the actual single cells. But we can also use another algorithm called MetaViper that actually integrates across all the networks that we've already built and automatically selects the best network. And so this is actually really cool. This paper has now just been accepted on Nature Communications, so should be coming out anytime. Basically, what we showed is that if I take the match network, for instance, if I have bulk tissue from gliomas, and I compare the master regulators that I get with, actually not the master regulator, this is the ability to identify the aberrant activity of proteins that have genetic alteration in the corresponding gene when that gene mutation is actually recurrent and occurs in, say, 5, 10, 30 of, say, the glioma patients. So if you actually look at using the correct glioma network, you get this. So you get a pretty good ability to recover the activity, especially for the mutations that are more recurrent. But if you actually use MetaViper, you do even better. So a MetaViper is eliminating, not using, in this particular case, the correct network. So eliminating the glioma network, using only the others. All right. So let's see what this does for single cells. So here's a FACS data for B cell and T cell, and these are melanoma cells. And so if you sort them according to CD3 and CD19 markers, the B cell would have high CD19, the T cell would have high CD3, and the melanoma cells, actually I don't know if these are melanoma, this is probably some other tumor, they would have no activity of either, no expression of either CD3 or CD19. So that's what you should see, all right? This is what you see when you do single cell gene expression profiles. Almost all the cells are either on the x-axis or on the y-axis because you get 0 reads either for CD3 or CD19. In fact, most of the cells are actually there in the 0, 0 point. They're one on top of each other. Right? So you've lost dramatic ability to recover the signal. If you actually use lineage marker, PAX5 and FOXP3, these are established lineage markers for these type of cells, and now you look at it's even worse because these are transcription factors at a very low level of expression. If you actually run MetaViper, you now completely recover your facts. So these are the T cells that have CD3, these are the B cells that have higher CD19, and these are the, in fact, in this case, our melanoma cells that have neither CD3 nor CD19. And if you do it with all the other markers, you get really spectacular segregation of these populations. And in fact, this is, for instance, the best set of markers for separating these three populations. So if you summarize this in a plot, what we did is the following. We said, okay, let's combine together all the profile from these cells and all the profile from these cells. So we get a kind of a synthetic bulk for each one. And let's look at what are the differentially expressed genes that are statistically significant. Let's take the top 100, and let's see how many of them are recapitulated when I just pick just one single cell from this population and compare it to the average of this, or I take just one single cell from this one and I compare it to the average of this. Okay? It's dismal. So this is the curve that you get, where basically about 20% of the cell may recover about 60% of the differentially expressed gene in the top 100. And so if you actually use Viper, MetaViper in this case, about 90% of the cells recapitulate about 60% of the gene. And on average, if you put a threshold here, say around 60% of the cell, more than 60% of the cell recover virtually every single one of the genes that you actually predicted. Yeah. So I'm going to go very quickly now because I'm running out of time. This just shows that we can do these very effectively with data that has actually been shown by FACS to have specific markers separating, for instance, monocytes and dendritic cells, separating in particular subpopulation like plasmacytoid and conventional dendritic cells by the appropriate marker like HLA, etc. This is what you get by gene expression analysis, which is basically almost impossible to decipher because, again, you have everything is on the X and Y axis. And so now what you can do is you can apply this to, for instance, study populations that are very difficult to isolate, like, for instance, tumor cancer, stem cell, in this case, initiating cells. So we FACS sorted using a CD49F antibody, which enriches for breast cancer stem cell. And now this is what you see. These are 5 patients. You can see by gene expression, they're spread all over. One of the things that Viper does, it actually now, when you apply Viper, they're now all on top of each other, so they're completely consistent. And now we can start looking at whether these make sense. For instance, let's look at CD49F, a beautiful gradient going from the fully differentiated cells to the cells that are less differentiated here. And so this is independent of the patient. You can now look at the biomarkers that have been identified for tumor stem cell, breast cancer stem cell, and each one of them gives you exactly the same type of profile. This, you would see absolutely nothing from gene expression, as I'll show you in the next slide, but you can even measure activity of microRNA. So this is a microRNA that is known to regulate BMI1, and you can see that the activity here is in the highly differentiated cells, no activity here in the more stem cell lineage. So these are the top, what you call master regulator or proteins that regulate the stem cell state, and you can see right next, this is the activity profile, this is the gene expression profile. So actually we're plotting the differential expression in yellow. So you can see that there is the pattern is somewhat conserved, but very few cells actually have expression, and you can actually get it. These are actually markers that are novel surface markers to isolate breast cancer stem cell, which are now currently validating. And the most exciting thing is that now you can apply exactly the same drugging methodology that we use for patients, you can apply to single cell. And so we can ask, among all the drugs that we actually profile in breast cancer cells, which one of them are predicted to kill or more likely differentiate the stem cell versus killing or differentiating the fully differentiated cells. And you see this beautiful pattern. These are about 400 drugs. These are all the taxanes. Blue means that they're predicted to work. And so all the taxanes, all the chemotoxic drugs are about here, and they all work only in the differentiated cells. So this is a gradient from stem to differentiated, and they do absolutely nothing for the more undifferentiated cells. Comparatively, these are the drugs that seem to work for potentially stem cell. They do absolutely nothing for differentiated tissue. Okay? And so we kind of looked at what these are, and I'm not going to bore you with the detail, but there's a tremendous amount of confirmation from prior studies. For instance, the topocyte, ivermectin is actually an analog, a chemical analog of a drug that was validated in a different study by Rick Lander, et cetera. So now what you can do is you can simply paint the activity of the drug on the single cell. You'll see they're all very active in this area, not active at all here. And now you can take a PDX model and ask, can I test this in a PDX? And so these are single cell from a patient transplant where we get exactly the same pattern. This is now done with the chromium. This is done with a different technology we developed with Jeremy Worley in the lab in collaboration with Peter Sims. And now you can see again, not only the same biomarkers, went one, notch one, but actually the activity of the cell completely, of the drugs completely recapitulated in the stem cell population, but not in the fully differentiated population. And therefore, this is something that we now test, and we're testing with Andrew Kang. So I'll stop here. I was going to thank a lot of people, starting from some of the people that really did the early work with us, including Antonio, Anna Latorella, Ricardo Lafava, Corey Abadeshen, Michael Shen, etc., and some of the people involved in some of the more recent work with Peter Sims, all the people that have helped us put together the N of 1, Andrew Kang, Eric Schwartz, Nina Hipshut, Mahesh Mansukami, Neil Suresh, Peter, and the International Net Consortium. The paper is now in press on Nature Genetics, accepted about a week ago. So, also wanted to thank the NIH funding sources, in particular my R35, the U1, and the most important one in the context of this symposium, U54 for the Cancer Assisted Biology Center. Thanks.
This is something probably talked about, but if the goal, as you described it, is targeting master regulators, and yet master regulators are not very druggable. So how do you explain your success with these drugs that you're using?
Yeah. That's a great question, and the answer is actually very simple. The same way that the master regulator integrate the effect of the genetics upstream, they also integrate the effect of the targeting of protein upstream. So the way we downregulate the master regulator is by hitting proteins, kinases, for instance, that are fundamental regulators of the tumor checkpoint. And of course, that's a problem because you are subject to the same level of potential failure that targeting a genetic alteration would have. For instance, you may develop mutation in the active site of those proteins. You may generate sort of alternative activation to other mutations that don't go through that fundamental protein center. But, for instance, the key protein that is used today for drugging the ABC subtype of the LDCL and also in CLL is called the Bruton tyrosine kinases, which never mutated any upstream of NF-kappa B because we don't know how to drug NF-kappa B. We're also working with several companies and also our research centers to develop both morpholinos and other type of tethered drugs that actually can drug master regulators. So the goal is eventually, now that we know we have an inventory of about 300 of them, and we know how they segregate each individual tumor subtype, is to actually go after the master regulator themselves.
Other questions? So one idea is that the, oh, sorry, go ahead. You have a question?
Yeah, how much?
So you mentioned that the de novo network inference from the single cell data was difficult. Is that surprising? I would imagine that a lot of the noise and gene expression data from cancers is cellular admixture, and if you peel that away, obviously the sparse data with single cell, but you should have a very clean.
You're absolutely right. In the sense that it's a balance. The problem with single cell is simply that there's a lot of dropouts, and therefore it's a very discrete number of counts. You get 0, 1, 2, 3. Fortunately enough, we've just developed an algorithm that uses exactly some kind of information theory-driven binning that allows us to deal with this kind of data. So this is a paper that we just submitted. And we use, so Ding in my lab was the guy that did all the work on the single cell, I forgot to mention him, but was actually the absolute mastermind behind all the work that we've done in single cell. Ding actually adapted what's called HP arachne to single cell. The value of single cells is that you isolate that context completely, so now you have a really pure context. In theory, you now generate networks that are individual specific, not even tumor specific, and you can actually generate networks that are specific to the P dendritic cells versus the C dendritic cells. So, in theory, you can actually get even more specific. You lose a little bit of the range of expression that you get when you go across the lineage. Because what we've seen is typically these regulatory networks are actually very conserved in the lineage. And so by not taking cells from different set of lineage, you may lose some of the dynamic range that you get on the expression. But it's not to go around because now you have a lot of single cells. So in the end, actually produces beautiful networks. They work better than the original networks from both tissues.
Other questions?
Yep.
Andrea, so you probably have thought about these things. So now they are coming every other week. There's a method for for denoising single-cell data. Just using zeros, using some kind of network structure, and then you start to do this thing. I mean, probably you've been checking about what's your opinion about what is the best way of dealing with this data?
So the editor and Nature Communication gave us a lot of grief because one of the reviewers asked exactly that question, so maybe it was you. And they said you have to compare this method. We said you can't really compare this method because there's no method that we know of that actually computes the activity of a protein from the expression of a lot of its targets. And so all the methods that are available today are methods that essentially impute the expression. For instance, Dana Pear's worked on a method called MAGIC that imputes by using the expression of the nearby cells. Unfortunately, the nearby cells are as hit hard by the dropout rate as that single cell. So we prefer to do it, and we showed obviously that we have to compare it to that method as well. We basically had to do it in a way that is completely, cell autonomous. Basically, it uses just the data from that single cell to make the imputation, as opposed to using the global context in which it is. But there's a lot of different methodologies that people use. The problem is that when you have zero read, for instance, you can use any kind of prior, Dirichlet prior on the probability density of the bulk, etc. But you still having a problem because when you see zero, you can't deconvolute zero. I mean, that's basically what you do. Oh, yeah, sorry, you had a question?
Well, yeah, I had a question, but so my question was, FOXM1 is a really key checkpoint in combination with various other key checkpoints. Yep. So do you think this provides the cell type specificity? I mean, I know FOXM1 CMPF is a very key prostate one. We showed that, but we together showed that. So what do you think about FOXM1 with other collaborators?
So we've looked at FOXM1, in several papers that we published. For instance, in follicular lymphoma progression to diffuse large B-cell lymphoma, FOXM1 actually cooperates with other genes like NF-κB and others. But the really surprising thing was that the exact tumor checkpoint, the NM33B, the FOXM1, CMPF, but the top 7 of the 8 genes are actually conserving about 30% of the tumors in TCGA. And that is the 30% with the absolute worst prognosis. They're all highly metastatic, highly migratory, and they all, and they're all tumor, and they're all immune evasive. So I think to some extent it was a surprise because we thought, as you did, that some of these modulators, some of these regulators were actually very specific to the cell type. Turns out that it is actually not the case. Now, of course, to drug them, you need to drug different things because what's upstream of FOXM1 CMPF in a, say, glioma, and I don't even know whether that's important glioma, is going to be very different from what's upstream in a prostate cancer cell. But if we had drugs to drug the master regulator directly, then that problem would go away.
I was thinking of kind of a basket trial for master regulators.
If we had the drugs to target a master regulator, yes, that would be fantastic.